Hello everyone, my name is Professor Sarah Hosking. I'm the Chief Executive Officer of the National Breast Cancer Foundation. It's my pleasure to welcome you to our webinar this morning with Professor Judy Garber, who joins us live from the Dana Farber Cancer Institute in Boston, USA, where I think I'm saying the time is about 8 p.m. in the evening. Professor Garber is the Director of Cancer Genetics and Prevention and also Professor of Medicine at Harvard University. She has an international reputation for her research into the genetics of breast cancer, and in particular, how risk factors that cause people to develop the disease can be used to predict cancer outcomes and even to reduce cancer risk into the future. I was very fortunate to meet Professor Garber earlier in the year, and when I heard her speak, I was absolutely sure that our NBCF community would really be very interested to hear the latest developments in this area of research. So I welcome Professor Judy Garber.
Hello. Can you hear me?
Yes, Judy, please I'll just get your slides up.
Great, thank you. It's a great pleasure to feel like I'm back in Australia, although I have to say that the weather in Boston has improved tremendously since I visited in April here. And this slide, which I hope you can all now see, was the first slide for what was the Connie Johnson lecture at the Garber Institute, in honor of Connie Johnson, who many of you may know. I didn't have the pleasure of meeting her, but she was clearly a remarkable woman and battled her breast cancer bravely and left a mark. So it was an honor to give a lecture in her name. So if we're okay, Hannah, let's go forward to the next slide. So these are my conflicts. I do some clinical trials in breast cancer with medications inhibitors, which are used in Australia as well, both in investigational uses in clinical trials, and there is now an approved drug in the United States, and I believe in Europe and Australia as well. The INFORM trial is a treatment study supported partially by one of the testing laboratories in the US, and Amber Genetics is another lab that we do research with.
Excuse me, Judy, sorry, I'll just put you pause for a moment. We're just having some audio problems.
Oh, I'm sorry.
That's all right, we'll just give it one moment.
And epidemiology for many years, many important trials, studies coming from Australia. And we know some things about what causes breast cancer, but we're still missing a lot of information. Next slide. Oh, I think we have a delay on the slides. Can you advance the slide, please? Great. So the kinds of risk factors that have been most powerful have included in the upper left the inherited risk, things like mutations in the BRCA1 or 2 genes. Breast cancer itself, women who have had a breast cancer or ductal carcinoma in situ have more risk of developing a second cancer than the first breast was exposed to, so was the second. The third figure is therapeutic radiation, so women who had treatment for diseases like Hodgkin's disease in childhood have an increased risk of breast cancer. On the bottom right is a slide that a pathologist might see about what breast cancers or what precancers can look like under the microscope. These are lesions like lobular carcinoma in situ or atypical hyperplasia. And at the bottom left are a series of mammogram views. So these, you have to imagine what one view of a mammogram might look like. And this is on a series of 6 different breasts from women who have more or less density. On the right, the whitest is the most dense, the higher the risk of breast cancer. Next slide, please. I think you'll have to click twice for this. So on the left in the first box are the fixed factors. So what are things we can't necessarily do anything about? Gender in general, age, no we try, hormonal factors, benign breast disease things that are found on biopsy, family history, history of prior breast cancer, as we said, radiation and mammographic density. On the right are things that are more modifiable. So exogenous hormones means hormone replacement, hormones you might take, potential control pills, although they have a very small effect, body mass index or weight, which is harder to control than we might like, diet, alcohol, of course, exercise, and some mammographic density is actually modifiable depending on hormonal factors. Next slide, please. This just shows that these various factors are different for different subtypes of breast cancer. So first, we divide breast cancer into whether it occurs pre or post menopause, so younger women or older women. And then on the next slide is whether the breast cancers are hormonally driven, the estrogen and progesterone receptor positive breast cancers, the female hormone receptors show that the tumors usually are sensitive to hormonal factors in the environment of the person, or estrogen and progesterone receptor negative, where hormones in the environment of the person don't seem to matter one way or the other. And if you look, most of what we know about what causes breast cancer related to the hormonally driven breast cancers. Early menses, or when your periods begin, whether or not you've had pregnancies and when the first one was, weight, at least partially because with more fat, we have higher hormone levels often, at least after menopause, oral contraceptives, and maybe family history. These are factors that affect estrogen receptor driven or estrogen driven tumors more than the so-called triple negative or hormone receptor negative tumors, and we're still learning more about them. Some of those are strongly related to family history. Next slide, please. So here's density again, and for a long time people wondered whether the pictures like the one to the right, the very dense mammogram, was more about masking a breast cancer. After all, it would be very hard to find a small breast tumor in a breast that looks like this. But as the groups in Canada in particular have studied, the denser a woman's breast is, the higher her breast cancer risk. And this is true among premenopausal women, some of whom don't have very dense breasts, although most young women do at least have some density, and in postmenopausal women, where after menopause, when hormone levels fall, most women's breasts become less dense. And those women whose breasts do not become less dense are the ones who have higher breast cancer risk. Now, but the observation nonetheless holds. And in the US, where women have regular screening mammograms, there are now many states that have laws that say that if women have very dense breasts, they need to be offered the possibility of additional screening modalities, so maybe ultrasound or MRI, to try to get around this problem of having such an obscure picture on the mammogram that the test itself is not as useful. Next slide, please. I don't exactly know how this came out this way, but it's just to remind us that people have tried very hard to make sense of all of these risk factors and have looked over the years at whether they should be added together or multiplied, or how to cope with multiple factors. And one of the main groups to work on this was the group led by Tyrer and Cusick, who are in the UK. They ran a large breast cancer prevention trial for women at increased risk, primarily based on family history. This was the first of the tamoxifen prevention trials, and this was also run in Australia. And from the group of women who participated in the study, they generated a model that's now available online, and you can barely see in the back that you can if you go to your browser, you can Google Tyrer Cusick calculator, and this will come up. You fill in all these risk factors, and you get something that looks like the center of this, a white screen with a family history, a pedigree in the middle, and a set of graphs at the bottom, and it will help to try to calculate breast cancer risk over time. So this is a useful tool that people use in assessing risk and counseling about risk. It's not perfect, and it gives a very accurate risk, but many groups try to use this to help women get a sense of how much risk they have compared to the general population. In this example, the red line is obviously much higher, much more risk than the blue line, the general population. But these tools at least exist to try to help us have a sense of how much risk we have. Next slide, please. This was the IBIS study, and I include this because, so this was 20 years ago now. In the early 1990s, there were 2 large clinical trials to try and see whether the medication tamoxifen that was used to treat breast cancer could prevent the disease in women who had never had breast cancer. And the reason that people thought this might be true was that women who had breast cancer and took tamoxifen not only had a reduction in their risk of having their breast cancer come back and an improvement in their survival after breast cancer, but they were less likely to get a second breast cancer. So this trial was done to see whether tamoxifen could prevent breast cancer. Not a perfect drug, nonetheless. Next slide, please. And what was found, so this actually is the result from the US version of the same trial. They were run really at the same time, but they were quite separate. And this was an attempt to show so on the left, you can see that among women in red who did not take tamoxifen compared to women in teal who did take tamoxifen, there was half as much breast cancer among the group that took tamoxifen. There was 2/3 as much breast cancer in women who, as much ductal carcinoma in situ in women who took tamoxifen, and there were fewer fractures in women who took tamoxifen. But on the risk side, there were endometrial cancer and there were blood clots and a few more strokes. So this was not a home run. It was important to show that we could prevent breast cancer, but the study also showed that women did not want to do this at such a high cost of other factors for their health. Next slide, please. One of the most important findings, though, study came years later. So the study was finished around 2002, but in 2015 they published the 20-year follow-up data. So this is breast cancer, and people have been so good about contact with the groups that ran their studies that we have 20-year data. And what the data showed was that women who managed to take 5 years of tamoxifen, and most often that was younger women who didn't have such side effects, but those women were still getting a protective effect of tamoxifen. Here, the placebo group is having more breast cancer developing all over these 20 years than the tamoxifen group. So this drug still worked after women stopped. That's pretty amazing. And for women for whom tamoxifen is a reasonable drug where the risks of serious side effects are lower, usually because they're younger and generally healthier, you get a very long-term effect of tamoxifen, and that's an important thing to keep in mind because I want to think about that as we think about other prevention interventions. Next slide, please. So who is at highest risk of breast cancer? Well, so women who have mutations in the BRCA1 and BRCA2 genes. And I'm sure in Australia, like the rest of the world, and in Australia where we all had, you all also heard about the Angelina Jolie declaration that she had surgery to avoid breast cancer because her family had a BRCA mutation. Other genes are on this slide. And the third category are those pre-malignant lesions on biopsy that we talked about, chest radiation, prior breast cancer. So it's the same list that we had before, but this gives you a little better sense of the amount of risk. These are enormous risks. In the general population, we're talking about risks in the range of 10 to 12% over a lifetime. These are higher risks, and these are things we have to think about dealing with for the women who have the. Next slide, please. I'm sorry, I can't change the slides. There we go. Well, so that's okay. So this slide shows how genetics has permeated our consciousness, and it has survived. Not all of these magazines still exist. And at the bottom is Angelina, who gave us some vocabulary with which we could discuss inherited risk. Next slide. And here is a pedigree. So pedigrees are diagrams of families. Everybody's doing Ancestry.com these days, I guess. So we all have seen pedigrees now. And you know that on families or pedigrees, circles are women and squares are men. This is by convention. That's always the same. And in this slide, where those circles that are colored in are kind of the person who's coming to medical attention at that time. So it's a young woman diagnosed at 32, and you can see that she has breast cancer on both sides of the family, but she has more than one breast cancer on her father's side, and it's her father because he is the square connected to her directly. And his sister had breast cancer at a very young age, 29, and ovarian cancer, and the line means she has passed away. So this family on the father's side is pretty classic for an inherited breast ovarian family, like the kind that might have a BRCA1 or 2 mutation. On the mom's side, mom had breast cancer too. Breast cancer is a common disease. It could have been for an inherited reason. It seems a little less likely because her mother and two sisters are older. And here's our patient whose sister at 29 must be concerned that she might be next. Now, families like this have existed forever, I suppose. Certainly for hundreds of years they've been recognized, and it was with the identification of genes like BRCA1 and 2, and now more recently additional genes that the field of cancer genetics was born, back in 1985. So it's been around for a while, and it's become more and more active. And I have to say that Australia has been an enormous contributor to breast cancer genetics because the country has been so well organized by the SIMBA consortium. Many of you, if you've had breast cancer or have had genetic evaluation, have had your work done through institutions that are connected in the very organized Australian healthcare system, the envy of the US certainly. And much of the day tonight comes either from, or today for you, from studies of the Australian group or coordinated by Australians for the rest of the world. Next slide, please. So why do we think that there's inherited susceptibility? So this goes back to a model developed by Alfred Knudsen and Louise Strong, two geneticists who basically did this as a mathematical model. But they looked at the example of childhood cancers and said, why is it that in some families, cancers cluster? Cancers they focused on were retinoblastoma, a tumor of the eye, and Wilms tumor, a tumor of the kidney. And they said, these are rare events in the population, but there are some families where multiple people have these rare tumors. How could that be true if they were genetic? And their hypothesis was that it would be possible in a tumor to have lost both copies of a particular gene, both the one that comes on the maternal chromosome and the one that comes from the father, the paternal chromosome. But someone could be born with one normal copy, altered copy, a mutation that came from either parent. So a mutation is a change in a gene that usually alters the function of the gene. Mostly it inactivates the gene. Sometimes it just damages the function of the gene. But the idea was that everybody with cancer that they acquire, so the person on the left who has just 2 green stars, that person in the course of his lifetime might have been exposed to cigarette smoke, a mutation that stayed with the cell in one copy of a gene, but that someone who was born with an altered copy or a mutation in one copy of the gene in all of their cells only had to get a second hit, something to happen to the partner gene in one cell for cancer to happen. And you can see that in the in the appearance of the green on top of the red in that individual. So I don't know if this is clear. Next slide. But the idea is that if you were born with one altered copy in all of your genes, that you would only have to acquire one, have to get 2 hits, something to knock out both copies of some gene in the course of their lifetime, and that would take longer. This is just a diagram showing the chromosomes, and in red along the chromosomes are the names of many cancer susceptibility genes. So when Knudsen and Strong worked this out, we didn't have molecular genetics. We had peas from Mendel, we had worms, we had flies, lots of organisms you might have studied in school. But now we had molecular testing, and now we know of over 100 genes that confer increased risk of cancer, different kinds of cancers. Can we get the next slide, please?
Come on.
No. Oh, there you go. Okay, so how good are we at figuring out genes? So this is just a diagram from Dr. Easton, who's in the UK, and basically he says, you know, we can figure out cancer risk that explains about half of all the genes we think are out there for breast cancer. So that doesn't mean that all breast cancers are genetic and we can only understand half. It means that of the genetic ones, we can only understand half. And how many are genetic? Well, that matters too, but somewhere probably around 25%. And the rest occur for all kinds of other reasons. Cancer, just to be clear, so cancer itself is a genetic disease. It's a disease of the genes. The genes in the cancer are altered. It changes the way those cells behave. They're not normal cells anymore. But all of those changes are acquired over a lifetime. No one is born with them. We're talking about conditions here where people are born with one altered gene and then acquire all the other genes on top of that. Next slide, please. So this is the ASCO version of genetics. In the cell over there on the left, there's the nucleus, the center of the cell that contains the genetic material. The genes are organized on chromosomes. We each have 23 pairs of chromosomes. If we're female, we have 2 X chromosomes that make us female, and the other 22 pairs. If we're male, we have an X and a Y, and the other 22 pairs. So when you get to the gene level, there are colors in there. That's because genes are made of nucleotides. There are 4 of them. It's an amazing system. 4 little letters of DNA code the entire genetic code in all of our chromosomes. The estimate is there are about 22,000 genes in humans. And the way genes work is that they give the blueprint to the cell to make proteins, and the proteins do all the work in the cells. So if your genes are correct, your proteins are correct, your cells do their job. If there's an error in one of those genes, then the protein won't be correct and it won't work right. And as I said, you can acquire these changes or you can be born with them. Next slide. So here's a picture of chromosomes. Chromosomes normally should look paired. They don't quite look like this. They don't come color-coded and they're not all the same size, but basically they're matched sets. And if you have a mutation in one copy of one gene, your chromosomes still basically look exactly the same. Next slide. But what kinds of mutations do you have? And this just reminds us that mutations or changes in these genes can be extremely subtle. So how we read those DNA letters in a 3-letter code. So here's an example. Wild type just means sort of the normal version. The big red dog was fun for, and you assume there's the rest of the sentence. You can insert one letter, and if you look there, that M is in the middle of dog. And basically it moves all the other DNA letters up. They're still the same, but now they're grouped funny. And that, you can see that the rest of that sentence doesn't make any sense. And you would argue that if this were a gene, the rest of that gene wouldn't make any sense, and the protein would come out with an error in it. It would be probably all that gobbledygook on the right would be decayed and the protein would be too short to do its job. And the deletion, same thing. We lost the OG of dog. Now it doesn't make any sense, so it's still not going to work. At the bottom, we have a very subtle change. We've gone from a red dog to a rad dog. So is that a subtle change or is that an important change? Some of them are important if they're in just the wrong place. Sometimes they're just human variation and they don't matter. And that's why underneath you see that these variants get classified as either pathogenic and obviously going to be important to that chromosome or that gene, or all the way to the right, benign, where we think that that's just a subtle change that doesn't matter. Next slide, please. We have all kinds of mistakes that the cells can make. You know, when your cells divide and reproduce themselves, which they do all the time, in your blood, in your hair, really in all of your body, every time the cell reproduces, it has to make its DNA perfectly, but sometimes it makes errors. This is an example of a sort of spell check error. If you look on the left, you have capital yellow T in the middle paired with C, because all of these pair A's and T's, C's and G's are correct. But in this one, the T is pairing with the C. And if you look at the top, that T got spliced out because it's a mistake. That C should be paired with the G. At the bottom, it didn't get spliced out, and that means that that mistake is going to go through with the cell every time it reproduces. So this is a spell check error, and this is commonly seen in colon cancer, not so much in breast cancer. Next slide, please. So that's cancer, and it has mutant genes. Little joke. They tell you it's humor, so you'll know that just in case. Next slide, please. On the next slide is an example of actually what those chromosomes look like. This is called chromosome painting, and this is a cancer cell from someone who has a BRCA2 mutation, and you can see that there are all kinds of amazing looking errors here. So these chromosomes don't quite look like X's like the others did. But maybe number 5, but they're not the same size. So this is what happens when there's a gene that's important in fixing errors in the DNA, which is what BRCA1 and BRCA2 do. And when you can't do that, the cancer cell is able to just accumulate all kinds of changes. No wonder it's a cancer cell and doesn't act like a normal cell. So that is the best example I know of how to think about these mistakes. It's actually amazing. Next slide, please. Thank you. So it turns out that all this DNA repair is complicated. No surprise. I mean, your body has to have ways of fixing errors in the DNA, and it turns out it has 5 pathways of doing that. And each of them has to be perfect for things to be, well, perfect, I guess. And I put that up not because you're expected to remember that, but you can imagine that when scientists first discovered BRCA1 and 2, and were then able to sort of figure out the pathways of fixing errors in the DNA, they also were able to then say, well, maybe some of these other genes are also part of the genes that confer susceptibility to breast cancer, or finding genes and then looking at these genes in families to figure out if there was cancer risk. And I don't think we've explained why BRCA1 and 2 are so important for breast and ovarian cancer, for example, and don't really confer risk of colon cancer or lung cancer, and other genes in these pathways give rise more to other cancers that we haven't explained. But at least we've got a pretty good catalog now of genes to try to sort out. Next slide, please. We know how to look for these genes, and we know some of the risk factors. How do we decide what to do? So I put this slide in because if you go to the doctor and you say, well, you know, I'm curious about my cancer risk, I'd like to have my genes tested, usually especially in countries like Australia, the doctor has to figure out whether you make the criteria. So the criteria are there because they want you to have a reasonable chance of having a positive test. If tests were completely free or cheap, they would probably make them more available. They certainly will because they're becoming less expensive. But you look for factors that might suggest that something genetic is going on, and you do that based on model. If all the cells in the body have one altered gene, that's why you can look in the blood for these genes, and you don't have to take biopsies of the breast or the ovary. And you can predict that if someone has these, that there might be more likely to have their cancers earlier in life because they don't need an extra hit. They only need one extra hit that can come early, or they might have more than one body organ that has cancer, or they might have something rare like breast cancer in men. So why would you test someone? Well, if they told you there's a mutation in the family, hopefully that would be a good reason. People who had breast cancer at unusual ages or had cancers more than one time in the breast or the ovary, male breast cancer, other clustering in families of breast or ovarian cancer at all. So data from Australia showed that among women with ovarian cancer, almost 20% would have a mutation. That made it possible to test everyone. Pancreatic cancers can be associated with these genes, triple negative breast cancers, and now some tumor analysis might lead you to think about testing for inherited cancer risk. Next slide, please. Thank you. It's coming, I can see it back there. Good. So, okay, for many years, we found two. Those were the first genes we learned about in the early, I guess in the mid-1990s. And then as people were walking down those pathways of DNA repair, trying to figure out about other genes, Dr. Mary Claire King, who had mapped the BRCA1 gene to begin with, said, you know, this is taking too long. We need to look for all these genes at once. And she did this by studying 360 women with ovarian and related cancers and independent of family history. And she looked at 12 DNA repair genes at the same time instead of one at a time, and found that 24% of these women had a mutation in some gene. And you can see from that circle over on the right from the pie chart that most of them had BRCA1 or 2 mutations, but others had mutations in others of these DNA repair genes. And that day, really, things changed. On the bottom, you can see that there were two sort of scary factors if you were a geneticist at that time. First, 30% of those women had no family history. Now, of course, women today have small families. There might not be other women, maybe just the mother, for example, all brothers otherwise, and you wouldn't have seen family history. So that was sobering. It reminded you that not everybody came with family history classically. And I told you women were supposed to be younger when they had these genes, but 37% of these women were over 60. And although I now think that's not so old anymore, I would say that that wasn't young by these criteria. Next slide. So here's a family, and now today, instead of looking for BRCA1 and 2, many people would test for multiple genes at the same time. And we would test even that young woman with breast cancer already, because we'd like to know for the rest of the family, was this genetic? Next slide, please. So those arrows just show that if that woman was, if that young woman does have a genetic risk, that there's implications, at least for the other women in the family. And of course, today we would know that, oh no, sorry, that we would know that many of those I'm sorry, somebody sent me a message on my phone now, I'm confused. Okay, so the arrows are to show other people for whom the implications of that testing are important. If that young woman has a mutation, do these other young women? And actually, it's not fair. It doesn't point to the men, but men have risk too, and men have children too. So we no longer think of this as only a problem for women at all. Next slide, please. Next slide, please. There we go. So this, if you are old enough to remember the Where's Waldo books, which I used with my children. Where's Waldo was a game where everybody looked like a human there from some period in history, and you were looking for Waldo. And if you hit the button one more time, you'll see Waldo at the top of the screen, or the top of the picture. Once you know the mutation in the family, then you can look just to see if that mutation is present or absent in family members. And if it is absent, that does not share the genetic risk in the family, though they still have the same family history. And if it's present, then obviously they have the same mutation. So this kind of testing can be definitive in a family where a mutation is known, usually much less expensive. We're not so sure it's always enough anymore, but it is certainly in a family with a clear mutation, this can be very helpful testing. So if you would just hit the advance button, first we'll see a circle come around Waldo, there he is. So obviously once you know where Waldo is, the next time he's much easier to find. Next slide, please. So on the top above the red line are the 7 genes that are the most connected to breast cancer at this time. There's BRCA1 and 2, P53, P10, CDH1, but you can see on the right that these are not just breast cancer genes. They all give risk of some other gene and sometimes of some other cancer, and sometimes those other cancers are even more important than breast cancer, if you can imagine. At the bottom are genes that are linked to breast cancer, but not strongly. So relative risk is a way of comparing risk against, for people who have an exposure here like a gene, against people who don't. So in BRCA1 and 2, the risks are about 11 times as high in people with a mutation compared to people without. P53 is even higher. Below the red line, the risk of breast cancer is somewhere between 2.5 and 3 times as much in people with mutations in those genes. Next slide, please. How much risk? So here's BRCA1 and 2, and this is the way we've thought about this for a long time. You can see that BRCA1 and 2 are not exactly the same. They're pretty similar for breast cancer. When you get to ovarian cancer, there's much more risk with BRCA1. When you get to prostate cancer, there's more risk with BRCA2. And these are higher risks, obviously, quite high, never 100%. Never 100%, and that's important because people feel like these things are inevitable, but they are not. And in every family, we find people who have mutations and not cancer. And they live long lives, they still don't get cancer. We need to know why that happens. Next slide, please. In the next slide, you'll see graphs. So these are from 2017. So these are the most recent data in a large paper published in the journal JAMA, the Journal of the American Medical Association. You might want to put your mute on. And in that, you can see that these are based now on large numbers of women and people with cancers, people with mutations. So SIMBA is an organization run from Australia to which all investigators around the world have contributed data on their individuals with mutations who have agreed to have themselves studied. Many of these have given blood samples and other samples over the years and have been tracked to see what happens to them, and you can see the risks of cancer. These are now very stable, and when we give people cancer risks based on these numbers, we are quite confident that they are good on average. They are not great for each individual, but they are for the population as a whole. They are based largely on people from Western countries. Now we are getting more data from testing being done in China, being done in India. Still, we have very little testing in Africa and almost none in Russia. Next slide. Could you put your mute on, whoever's making noise? Thank you. So how do we take care of people whose risk we have? So now we know that their risks are so much higher. What do we do for them? So if you will hit the advance button a couple times, it'll put up some red X's. There you go. I guess you should back up. Thank you. Just to show, so one of the first questions was, can we find cancers early if we look? Are mammograms enough? And you can see the sensitivity of mammograms. So the chance of finding something if it's there with a mammogram in women under 50 and over 50 is not very good, only 40%. In general, in the population, we say it's 85% able to find something if it's there. So mammograms, not so good. Breast MRIs, much better, 85%. If you add them together, even better. Not 100%, but more than 90%. And that's why most women with mutations are offered screening with mammograms and MRIs. These studies were all done with everything done once a year on the same day, but now most people alternate, do them every 6 months. Next slide, please. Risk-reducing salpingo-oophorectomy. So one problem we have is that ovarian cancer is at least as big a challenge as breast cancer for women with mutations. And the problem there was we have no early detection that works. Ultrasounds, CA-125 blood tests, not very good. And we can show, though, if we remove the ovaries, that we will not only prevent the development of ovarian cancer, but we will improve survival. So women are less likely to die of ovarian cancer, 80% less likely. So there is RR.2. It's reduced by 80%, 1 minus that 0.2. It does reduce breast cancer risk in women with BRCA2 mutations, and it reduces mortality from other causes as well. So it's a pretty amazing intervention. It is probably the main reason we need to find people with mutations, because we can spare them from a cancer that most often is not curable. Next slide, please. But the side effect, of course, is that we make women menopausal at young ages. And if they still keep their breast tissue, they're worried about taking hormones. Next slide, please. Actually, this is the design of a study being done right now in the US, asking the question, can you prevent ovarian by just taking out the fallopian tubes, those tubes stuck to the ends of the uterus that are the way that the egg gets transferred to the uterus for pregnancy. But at the end of the fallopian tubes are the things called the fimbria. They're like those long fingers that sort of move to make currents to bring the egg from the ovary to the uterus. Anyway, the thought is that that many ovarian cancers actually develop in the fallopian tube first, and then the cells fall onto the surface of the ovary. And perhaps if you remove the fallopian tubes only and leave the ovaries behind, you can prevent ovarian cancer and not make women menopausal. But it's an important question for women around the world. Next slide, please. Here's data from a tool called Ask2Me. This is a Harvard tool from two guys at Harvard, Giovanni Parmigiani and Kevin Hughes. Ask2Me stands for All Syndromes Known to Man Evaluator, a very Harvard title. But you can Google Ask2Me and you can put in a gene and the age of a person, and it will give you the risk of the different cancers associated with mutations in that gene, not only as a graph, but also as a table to help you think about risk time. This is an important thing because we can give you these lifetime risk numbers that are overwhelming, but they may have a very different impact in women depending on the age at which they learn they have a mutation. So if you're a 30-year-old woman who has learned she has a BRCA1 mutation, you have a lot of risk in front of you, and a little bit every year, still a little bit each year. You can see that on the graphs. The risk is not 80% each year. It's a total of 80% over a lifetime, a small amount each year. But if you're a 60-year-old woman who has not had cancer, perhaps you were tested because a relative was diagnosed and you find you have a mutation, you have lived through a lot of that risk. You don't have 80% of your risk in front of you, and that might also affect your decisions about care. So these sorts of models or these ways of expressing risk might be helpful in thinking about that. No matter when you learn you have a mutation, we're likely to want you to take out the ovaries because the same problem of detection still exists, but other management issues like breast surgery is something that would be affected by this, we think. Next slide. This is the PROMPT study that is a way for people, particularly in the US, to contribute data to studies, even if you're tested by your doctor in a small town, not in a major cancer center. And we're just trying to accelerate research using things we've learned from Australians. Next slide, please. We do know these genes, the ones that were at the bottom of that table that only gave 2- or 3-fold risk, they also give increased risk. The challenge is, how do we help patients take care of those? We shouldn't treat all mutations the same when we know that the risks are not the same. So this is a table that we created for physicians to try to help them think about screening a little bit differently. For BRCA1 and 2, we start screening women at age 25 because they can develop cancers as young as 30. For these genes, the cancers are more likely to occur later, so we think the screening should start later too. Nobody needs extra mammograms. Next slide, please. This is just to remind me that you may know, certainly if you've had cancer, that these days people are analyzing the genes in the tumors, not doing that to see if they develop because of inheritance. They're mostly doing that to look for changes in the tumor DNA that could be targeted by particular therapies. But when you look at the tumor DNA, you may find mutations like BRCA1 and 2 that should give you a clue that they might have developed because of inherited risk. Next slide, please. This is the diagram from a study done in New York City at Memorial Sloan Kettering Cancer Center, where they sequenced the tumors, genetically sequenced the DNA of tumors and blood from 1,500 people. So this just shows you where technology has gone. And what they learned in this study was that by studying just 61 genes, that 17% of people who were treated at Memorial Sloan Kettering that year for cancer, an inherited risk. Not all that risk matched up with their tumor. Sometimes they had a colon cancer and a BRCA1 mutation, but they did find mutations in that many. And I think that's going to change care, and most people with cancer will be studied in the future to see if their cancer developed because of a gene, or if there's a gene that might benefit the rest of the family. Next slide. Now, we can skip this slide. Although these are data from Mandy Ballinger, who's in Australia, in Sydney, and they looked at people with sarcoma, a rare kind of tumor, and they found that almost half of some genetic contribution to where these tumors came from. So it's not the same for every cancer. Next slide. This is Li-Fraumeni syndrome, a syndrome associated with mutations in a specific gene called TP53, and it includes very early breast cancer, but there are also other cancers, particularly tumors of the adrenal glands and a connective tissue in the body, and childhood cancers. It makes it a particularly challenging condition. Next slide. Probably should have taken these slides out. It's a rare condition, but it does happen. And because it can attack many different parts of the body, the clear management issue has been to find cancers early. Next slide, please. So there was a big study done in Canada that showed that we could use a new technology called whole body MRI. So this is MRI screening, but it's from, in our case, we do it from the head to the knees, and we look at the whole body without radiation. MRIs are based on magnets, and that's important for these people because radiation seems to cause their cancer. So this showed that people who were screened, those in the red line, lived longer and did better than people who were not screened. And that might be true, but one of them certainly is that the technology actually works. Next slide. You can skip that slide, thanks. It just means that if you have a cancer syndrome that's got a lot of cancers, you have to have a lot of different screening. So we've seen this family before. Next slide. So one of the things we've had to think about, or were able to think about once we understood what BRCA1 and 2 did, was whether there was some implication of having a tumor that developed because of a gene in how that gene behaved, or how that tumor would behave. Was there specific biology that we could exploit as a way to treat these tumors more specifically and more effectively. So one of the things you can see, if you have a DNA repair gene or a gene whose job is to put those chromosomes back together perfectly and they're not working, that what you might see are errors in the DNA. So if you look on the right in the BRCA1 intact cell, you can see all those things that look like those chromosome pictures, except in the bottom right there's what looks like a cross. And if you look on the left, underneath the word deficient, there's another one. So these are two cells, one in someone with a BRCA1 mutation, one in a cell that's BRCA1 was okay. There was another error in the DNA. If the DNA, if you can imagine those chromosomes, so both strands are separated and they come back together in a funny way, you would get chromosomes that look like that. So this just tells you that you can really confuse your chromosomes if your BRCA1 is not working well. Next slide. That's in the tumor. So people thought, okay, if BRCA1 and 2 can cause these kinds of breaks in both strands of the DNA, this would be called DNA cross-linking. And there is a chemotherapy drug that causes cross-linked errors in the DNA, and that drug is platinum. That's not a drug we have used in breast cancer very much. We've used it for ovarian cancer, certainly, for testis cancer, for lung cancer, for other cancers, not so much for breast cancer. But here was the suggestion that if you had a BRCA1 mutation, perhaps platinum would be just the right chemotherapy to use because it would cause errors in the tumor DNA that your body could not, that the tumor couldn't fix because it was missing its BRCA1. In normal cells, in that person, there'd be one missing BRCA1, but having one normal seems to be enough for your cells to do everything normally, for people to grow up normally, have children, do everything. But in the tumor, if you lose the normal copy now, there's no functioning BRCA1, and these cells can't fix those errors. And that's good because you want those cells not to fix those errors and to die because of the tumor chemotherapy. So here's a study where people were given platinum before surgery. They did, this was neoadjuvant treatment. You may have heard that word before. They had platinum chemotherapy and then went to surgery, and in 61% of those women, when they got to surgery, there was no tumor left. It's pretty impressive because this was one drug. Usually, as you know, for chemotherapy, we like combinations, and we do that to make them more powerful. Next slide. But this is an old slide. This is from Israel in a study that looked at women with BRCA1 and 2 mutations. And basically what it showed was that women, I mean, we've all worried, were these tumors more aggressive? Would people do worse because they had inherited cancer? And what this showed was that as long as people got the right treatment, chemotherapy or hormonal therapy, that they did just as well as people who did not have a BRCA1 or 2 mutation. And this was old-fashioned chemotherapy. This was CMF. And yet people did just as well. Next slide. So this led to the discovery of a new drug called a PARP inhibitor. These drugs that target errors in the DNA. Again, you get tired of hearing about DNA repair, but it's BRCA1 and 2, that's what they do. And people thought, well, this is another drug that interferes with fixing errors in the DNA. Let's look for people whose tumors already have trouble in that direction and see if we can make it even worse for the tumor. And that's where they added these PARP inhibitors. Next slide. This is the sort of diagram of thinking about this. There are multiple DNA repair pathways. If you have a BRCA1 mutation, you've lost one leg of the table in your tumor already. And here comes the PARP inhibitor. It knocks out another leg and the table can't sustain itself in a vertical place, and the tumor dies or the table falls. Next slide. And that's what happened. So here is a diagram of the first study ever done where that for people to participate, the eligibility, you had to have a BRCA1 or 2 mutation. On the left, on the top, is a CAT scan. If you've never seen one, those black things are the lungs. The big blob in the center is the heart and the great vessels. And in the back part, you can kind of get the sense of the vertebral body, the spine. And there are some things circled in yellow. Those are tumor nodules in the lung. But if you look over on the right side, those tumor nodules are gone. Everything else remains. And that was what happened for some people with those PARP inhibitors. On the diagram below, you can see that on the left, those 3 little rectangles above the 0, those were people whose tumors did not shrink with tumor, but everybody else's did. They call this a waterfall plot. And it shows you that people did respond to therapy. And these were people who'd had a lot of other treatment first, but then they got this experimental drug at the time, and their tumors did shrink. Now, nobody's tumor disappeared. No one was cured by this therapy, but it did show activity, and that led to a lot of subsequent work in treatment of people with mutations. Now, for ovarian cancer, somehow it doesn't matter if you have a mutation or not, PARP inhibitors work, but in breast cancer, they really only work in people who have inherited risk. Next slide. Yep, this just shows that they're very effective, especially in ovary cancer, but also in breast. Next slide. This led to a study that's still ongoing, where people who have a BRCA1 or 2 mutation get all of their treatment first, either their surgery and then chemotherapy, or their chemotherapy and then surgery, or hormonal therapy, radiation, whatever they're going to get. And at the end, they're randomized. Half will receive the PARP inhibitor for a year. These are pills. You take 1 a day. Or they will have a placebo. It's the only way to figure out whether the PARP inhibitor adds to the treatment effect or the benefit for these, or does not. Does it have more side effects than benefit, or does it have more benefit than side effects? And the study has been going on now for a few years, and it will finish accruing patients, we think, by next year, because people have been going on study. It is active in Australia. Next slide. It's called the Olympia study. This is just to remind me that in another study using the same drug, olaparib, in people who had already had metastatic disease, this is all of the sites that were active. There are almost as many investigators as patients on these trials. These are rare, relatively uncommon patients, partly because people generally do well, especially if they've been in screening and have small tumors when they're found. But it just shows you it's a lot of people working together to get this work done. Next slide. This is just to remind me to tell you that there are studies now raising awareness, really, that mutations can develop in people, or can occur. People with mutations can get pancreatic cancer or prostate cancer, in addition to breast or ovarian cancer, and that these drugs work in patients as well. These are called basket trials, where to be eligible, you just have to have a mutation, not a particular cancer, which is certainly a different way of thinking than most of us use caring for our patients most of the time. Next slide, please. This is just showing that it works in prostate cancer. Next slide. See, it's almost 9 o'clock. Well, it's almost your time. This is data from an Australian investigator, Jeff Lindeman, in Melbourne, who has shown that a new molecule, rank ligand, may have something to do with the way breast cancers develop in women with BRCA1 mutations. There is a drug that targets rank ligand called denosumab that's used for treating bone health or osteoporosis, and it is going to be see if we can prevent or delay breast cancer development in women with BRCA1 mutations. We're very excited about this, and it's going to start very soon. Next slide. This is just the next slide. It's the trial design. It is randomized again to either the active drug or the placebo, and neither women nor their doctors will know which they're on, so we're going to need brave people to participate in this, but hopefully this will be an important study that may make a difference and make it so women don't have to think about surgery to reduce their risk, or at least can delay it. Next slide, please. Next slide, please. That's only a problem in the US, where you have to worry about patenting. This is just to remind you that people do genetic testing now all different ways. Next, you can hit it one more time. In the US, you can get genetic testing without your doctor. Next slide. We have a study for the Jewish community. We think now that it's important enough for testing that where mutations are 1 in 40 instead of 1 in 400 in the population, that people might be more interested in testing. Next slide. This is a study that's different mostly because, next slide, it's all done on your iPhone. It's got some videos that are animated that teach you about genetics, and then You have to go get your blood drawn and you get a phone call with the results. Next slide, please. This is not to advertise, this is Australia, you're not going to do this, but the goal is to show that genetics is becoming more accessible to the population at large, but still, hopefully in a responsible way. There are benefits to genetic testing and there are risks. It is not for everyone, or it's at least not for everyone at the same time in their lives. And all data that have been so carefully collected in the US and in Australia, studying people, have shown that people do best with genetic information when they got tested because they were ready themselves to be tested and not to make someone else happy. There are benefits for people who don't like uncertainty, who want to know their risk, sometimes to make medical decisions, but there can be family issues and personal issues that come from this, so we want people to make informed decisions about genetic information. But it is powerful information, and hopefully I have convinced you that there are many things you can do with this information to try to keep people safe and healthy. Next slide. We're almost done, I promise. We're done. So this is genetics. You got to figure out the risks and benefits of what you, and I think there are some questions, and I'd be glad to stay on a few minutes and answer some of them if I can do that. And I'm sorry that I took so long to get through the talk, but I guess we added a few minutes for the slides.
Thank you, Judy. Perhaps we might just have time for one question. I'll read that out to you now. I am a BRCA1 with a VUS. I am 4 years post-treatment for stage 3B breast cancer and 18 months post-surgery for an aggressive prostate cancer. My mum died aged 40 of breast cancer. Would it be fair to say I have a genetic propensity to cancers, and given that I have a VUS, should I advise my siblings and adult children to get genetic testing?
Good question. So what's a VUS? A VUS is a variant of uncertain significance. That's like the red dog and the rad dog. How do we know whether that's important or not? So I would say that for this man, I suppose, who's had breast and cancer and had a VUS and BRCA1, that you may have a genetic risk, but you can't assume that that variant of uncertain significance is the culprit. And that's a problem, because if you test your relatives, whether they do or don't share the VUS, you don't really know if that is a mutation. You're not sure what that is. I think one question is whether you yourself have had more expanded genetic testing. Perhaps it wasn't BRCA1 or 2, but one of the other genes we talked about tonight. And if you have not had that testing, you might want to do that so you can look for the mutation that would benefit your relatives. And if you have more expanded testing and they don't find a mutation, then although it certainly seems likely that there's something that explains the family, right now there's no way to test the relatives to see if they share the risk. And you will have to wait till more is done to find the other genes that predispose to breast and prostate cancer. And I'm sorry that I ran so long. If we can't answer other questions, if people send them into you, Hannah, I can try to answer them online if you can post them somewhere, if that would work.
Okay, that would be excellent. Thank you very much, Judy, for your excellent talk, and thank you for everyone for attending. And that concludes our webinar this morning.